Hello, my name is Campanella, and today I'm going to be presenting on Phylum Cycliophora. So, the phylogenetic position of Phylum Cycliophora is still uncertain. Right now, they have them under clade polyzoa, and they have them most closely related to the Anthropocotta and the bryozoans. They are determined to be protostomes. Their free-living larval stage represents a trochophore larval stage. So basically, it's uncertain because their downward feeding motion originally had scientists thinking that they were mostly closely related to the rotifers. So then they looked at their inner cuticle, and it was very similar to a nematode and a gastrorhynchus, so they weren't quite sure where to put them there. But then, after looking at their life cycles and seeing their inner budding, they decided to put them next to the Anthropocotta. The discovery of Cycliophora. They are recently discovered microscopic marine animal. They were discovered in 1995 by Danish researchers Reinhard Christensen and Peter Funch. And there are two known species. So there's Symbion pandora and Symbion americanus. And there's also one species that has been undescribed. A general overview, they are commensal with lobsters. They live in the mouth parts of the lobsters. You can mostly find them on the maxillae and maxillipeds. So the three different species are found on three different species of lobsters. The Symbion pandora is found on the Norwegian lobster. The Symbion americanus is found on the American lobster, and the undescribed species is found on the European lobster. This is a general overview of the body form of the Cycliophora. So right here you have the adhesive disc. This is where they stick to the lobster with their mouth. And then here's the internal bud. This is how they discovered that they were closely related to the Anthropocottas because of the way it's formed. And then right here you have the gut. So it is a U-shaped gut. And basically what happens is they catch the food of the cilia. Cilia brings the food down to the buccal funnel. Buccal funnel makes it's a downward feeding motion. You also have the cerebral ganglion. That is their brain, or what is considered their brain. Their nervous system is not quite known yet. It's still being researched, so there's still a lot to know. And here you have the esophagus and the anus. So this is the anterior side of the species, and this is the posterior side. Right here, you have the dwarf male and the sperm packet inside the dwarf male, and this is basically how they sexually reproduce. And this is the host ceteris, which is what they attach to. So they have a very complex life cycle. They have 6 different stages, and they are thought to be the most complicated of the metazoans. So a couple of their stages is the dwarf male stage, which is pretty unique to the Cycliophorans. They have the metagenic stage, so they alternate between sexual and asexual reproduction. And then they also have a multiple larval stage. So they have Pandora larva, corduroid larva, and Prometheus larva. So this is a picture of their general life cycle. So right here, 1 through 3, you have the corduroid larva, which is in its free living stage. So basically what it does is it settles onto its host, and then it comes over here and it generates into an asexual phase. And the buccal funnel forms, and it starts its feeding stages, and it's still growing, and as it's growing, its inner bud forms. So as its inner bud forms, the buccal funnel is regenerated and replaced. And then it moves on to over here, where once it reaches maturity, it turns into a Pandora larva. And then the Pandora larva escapes and redoes this cycle multiple times, creating multiple Pandora larvas. So they find that most Cycliophora are in their asexual phase. And they're not sure why yet, but they tend to see more Cycliophora in their sexual phase when the lobster had just molted. So getting into their sexual phase, it starts over here. So it forms the dwarf male and its feeding stage, and then it detaches itself, and in here this is where the female starts to form. So over here, the male settles and it keeps growing as it's feeding, and then the female escapes from the male, carrying an embryo, and then it comes over here and attaches with its posterior side. So since it's attached to its posterior side, it's secreting a lot of glands, and basically the cilia, they degenerate and terminate, and they go away. So then the female releases itself, leaving an embryo, and the choroid larva escapes, the female dies, and the process is done over and over again. So there's a study done on Symbion pandora and Tritichelophala. So these are Cycliophoras and those are bryozoans. Basically, the feeding biology was study of these two species, and it was compared by liquid scintillation counting. And liquid scintillation counting is when they take radioactive isotopes and they inject it into the food and basically count how much food each species is eating. So the types of food they used were algae and muscle particles. So the Cycliophora did not respond to the algae, they only responded to the muscle particles, but the bryozoa responded to both. So this showed why the Cycliophorans only live on the mouth parts of the lobster, and the bryozoans can live on multiple parts of the lobster. And then the Cycliophorans obviously rely heavily on their host's food. Thank you for listening today. This was a very interesting species to learn about. There's still so much research to be done about them. There's not much known yet, and I hope to learn more about it, hear more about it in the future. And thank you.